I'd like to talk briefly about why and how we think that metabolomics could contribute to better therapies. In order to answer that question, we need to look where there is a medical need. Which issues could metabolomics address in order to improve patient stratification? In our opinion, there's 3 areas where metabolomics could really be helpful: non-response, drugs becoming ineffective, and a general lack of actionable biomarkers. Let's go through them one by one. Non-response to pharmacological therapy is an issue that affects essentially all areas of pharmacological therapy. Even for metformin, it is estimated that 1/3 of patients do not actually have any clinical benefit. We think that metabolomics can be really helpful here because it provides a means of stratification based on the biochemical phenotype. In addition, it is much more informative than routine clinical biochemistry. This could be really impactful in clinical trials. If you have a good drug that still has a substantial percentage of non-responders, it might mean that the primary endpoint is not being met, although a substantial number of patients would actually benefit from a drug. Annually, 5 to 10% of diabetes patients need to change therapy, which is probably due to progression rather than any resistance mechanisms coming up. Loss of therapeutic response is also frequent in other indications, for example, depression or anti-inflammatory therapy in inflammatory bowel diseases. It is, of course, well known to be an issue in targeted cancer therapies. Genetics is often not very helpful in those settings because the genotype of the patient doesn't change. On the other hand, metabolomics can cover the dynamics of disease across the patient journey. In other words, if there is progression or if there is resistance coming up, you'll see it in the metabolome. Finally, for almost all drugs, there is no means of selecting the right medication. Cancer therapy is, of course, the rare exception here. Co-medication, nutrition, probiotic therapy, or lifestyle recommendations are largely based on trial and error rather than on a sound knowledge about the mode of action and how the patient is actually going to benefit from those. We think that metabolomics can contribute to better therapeutic choices and better patient management based on the biochemical phenotype, as well as a deep understanding of the biochemical mode of actions of interventions, which you could match with the molecular phenotype of the patient. If we look at biomarker research in pharmaceutical R&D today, we'll see that often the biomarker of choice is target expression or other biomarkers that are directly related to the mode of action of the drug. However, it is far from being the only factor that determines whether and how a patient is going to respond. Metabolism is certainly playing a role here. If we look at a typical clinical response profile, even in biomarker selected populations, you'll have variability with responders and non-responders in the cohort. If you look more closely, you'll be able to identify subgroups that respond very poorly to a therapy because there's something that suppresses the response. This is well known for antibiotics in an immunotherapy setting, for example. On the other hand, of course, there will be subsegments of patients who respond more favorably than the average patient, probably because there's something that boosts their response, which could be immune modulating therapy in immuno-oncology. If we understand on a molecular metabolic basis how response suppression and boosting response work, we could harness it in patient care, and we'll show you on the next slide how this could look like. What you see here is one example of biomarker research based on Biocrates technology. It's a study out of Heidelberg where patients with urological cancers have received immune checkpoint inhibitors. At baseline, the researchers looked whether metabolic profiles predict who was going to respond and who was not going to respond. And they identified certain lipids that contain very long chain fatty acids, together with age, did pretty well in determining whether someone was going to respond or not. Interestingly, the authors suggested that supplementing those very long chain fatty acid containing lipids could be used as immune sensitizer and probably promote cancer immunotherapy success. On a mechanistic level, very long chain fatty acids are fatty acids with 22 or more carbon atoms, and they probably promote peroxisomal function and thus anti-tumor immunity. Supplementing patients with those lipids would actually be fairly easy because they are contained in fish or fish oil. So the hope would really be to turn a predicted non-responder into a responder with a simple nutritional supportive therapy. On a more general level, we believe that metabolomics could and should be used as a performance companion diagnostics tool that stratifies patients not by target expression, but by how they are going to respond to a therapy. There's 4 questions that we think could be asked and answered with metabolomics. First of all, does a specific drug work in a patient? This is what's depicted here in the middle. Can metabolic profiles predict response or non-response or toxicity? Second, which drug fits a patient's molecular phenotype best, and also which co-medication or supportive therapy could enhance response. So ultimately, this would enable clinicians to decide whether a certain therapy should be started, whether it should be started but supportive therapy considered to enhance response, or whether a different course of therapy should be selected. The 4th area is metabolic profiles can be acquired pretty conveniently and repeatedly over time in order to enable treatment monitoring. Here's some of the publications that have been used in the previous slides. I'd like to show you one more example, again in the immunotherapy space, more specifically a study out of Innsbruck that looked at immune modulatory pathways and metabolome in general and immunotherapy resistance. First of all, the scientists looked whether there are changes in the metabolome between healthy controls and lung cancer patients, and unsurprisingly, there were pretty vast metabolic changes in those patients. Changes have also been identified between baseline and at progression, which suggests that really metabolic profiles could be beneficial in treatment monitoring. And most importantly, low baseline tryptophan levels have been found to be associated with shorter progression-free survival. That's what's depicted on the left side here. So again, the authors have identified a signal that indicates primary resistance to immunotherapy. This already leads me to the summary. We believe that metabolic profiles as performed in CDX would be a great extension of classical biomarker strategies. Metabolomics can help with therapy stratification according to comprehensive biochemical information, as opposed to the reductionist approaches that are common today. Metabolomics could also enable hypothesis-driven selection of supportive care, such as co-medication, nutritional, or lifestyle recommendations. And ultimately, we think that this will lead to improved success rates in clinical trials and also improved patient outcomes. With that, thank you for your attention.